Back.
All right, so tonight we're going to talk about nucleic acids. There are two types of nucleic acids. Nucleic acids are polymers made up of what are called nucleotides. And the two types we have are DNA and RNA, which we've already looked at previously a little bit. DNA is deoxyribonucleic acid. DNA is found mostly in a cell's nucleus. It is passed between cells during cell division, and it controls many functions within the cell.
I'm not seeing the slides.
Oh, I'm sorry. I did everything but resume sharing. All right, can you see them now?
Thank you, yeah.
RNA is ribonucleic acid. It's found in all parts of the cell, not just the nucleus. And its principal function is the synthesis of proteins, as we will see. So nucleotides have three components. There's a phosphate, a sugar, and then a base, all connected together. This makes up a nucleotide. And so the pentose sugar could be either ribose or deoxyribose. DNA is going to have the deoxyribose, and RNA is going to have the ribose sugars. And most of the bases are nitrogen containing, and they're derivatives of pyrimidine and purine, as we will see in a moment. And then we also have that phosphate component. So here you can see it's color coordinated. We've got this yellow section is the phosphate. It is attached to the left side of the sugar. And then we have this base, which is attached to the right side of the sugar. And let's see here. I'm trying to remember why I have these in here. I think just because this is ribose, because it has the oxygen, the deoxyribose, there is no oxygen on this functional group. And that's the difference. And these are the two parent compounds. Pyrimidine is a heterocyclic benzene ring with two nitrogens in it. And purine has that pyrimidine, but it also has a 5-membered ring with two nitrogens in it as well. You don't have to memorize these structures, by the way. So the derivatives of purine are adenine and guanine. So you can see both of these have that fused ring structure like purine. The difference here is that you've got a double bond oxygen on this one. You've got an additional amino group out here for guanine. And for adenine, you have an additional amino group up here. Pyrimidine has thymine, cytosine, and uracil are all derivatives. You can see that they have a single ring structure like pyrimidine. This one has a double bond O in it and a methyl group here and another double bond O over here. Cytosine has an amino group and a carbonyl group here. Uracil has your two carbonyl groups on it. And then the other thing that we can denote here is that all three of these, adenine, guanine, and cytosine, are in both DNA and RNA. These are abbreviated A, G, and C. But only DNA has thymine, abbreviated T, and only RNA has uracil, which is denoted with a U. So RNA can have A, C, G, and U, but DNA will only have A, C, G, and T. In the pyrimidine-based nucleic acids, again, you have this phosphate group. It is bound to your sugar on the 5th carbon, the C5 location, and the base is connected between the N1, the nitrogen in the ring, and the 1st carbon in this sugar. In the purine based, it's a little bit different. This phosphate is still attached to the 5th carbon, but here it's not the primary nitrogen, it is the 9th nitrogen on this ring that gets attached to that 1st carbon. So this is called a C1N9 bond, whereas this one's called an N1 to C1 bond. How do you name these guys? Well, it ends in 5-monophosphate because they all have this monophosphate attached to that 5th carbon. And then if the base, if it is a purine base, it gets osine as its ending. If it's a pyrimidine base, it gets idine as its ending. And then deoxy gets prefixed onto it if it's a deoxyribose. So our possible nucleotides, remember we just said A, G, C, and T are the possible primary bases on a DNA. So adenine would be deoxyadenosine 5-monophosphate, and it gets abbreviated as DAMP. Guanine, this is also a deoxyribose. Its name is deoxyguanosine 5-monophosphate, and it is denoted as DGMP, this D being that it's deoxy. And then with the RNA nucleotides, you can have A, G, C, and U. And so you just lose the deoxy on the prefix. And so the adenine-based one will be adenosine 5-monophosphate, AMP, it's just without the little D in front of it because it's RNA. Guanosine 5-monophosphate, GMP, so forth and so on. Any questions there? So like we said, we have RNA and DNA are made up of this sequence of nucleotides. So we're going to have a nucleotide with a phosphate sugar and base attached to another nucleotide with a phosphate sugar and base to another one, so forth and so on. This is a polymer of nucleotides. In DNA, the sugars are going to be deoxyribose, and in RNA, the sugars are going to be riboses. So the way that this is shaped is that how these chain together is these are called 3,5 phosphodiester linkages. And the reason for that is it's the phosphate that connects the two sugars and forms this chain. So we have a base here. In this example, it's adenine. We have our this is deoxyribose. It doesn't have the oxygen right here. And then we have a phosphate group that connects to that 5th carbon, but it also connects to the 3rd carbon of the next nucleotide. And that's why this is called a 3,5 phosphodiester linkage. And it's phosphodiester because this is like a phosphorus ester. So remember, ester is RCOOR. This is like RPOOR. This is like a phosphorus version of that. And this just continues. So it's got a 3,5 linkage between these sugars. Here we have cytosine hanging off of this, for example. You've got your sugar, and then there's a 3,5 connection through the phosphate to the next nucleotide, which is guanine, and so forth and so on. Here's thymine, and this could go on, you know, however long, hundreds and thousands of these in chains. You will always have a 5' and a 3' because you're going to have eventually a third carbon that is not connected anywhere, and you're going to have a 5th carbon that's not connected anywhere up here. Any questions there at all? You guys have probably had some of this stuff before in biology and things like that, right? So this may be a little bit of review for you all. I haven't had biology since 9th grade in high school, so you probably even know more about this stuff than I do. So these strands, remember we said there's going to be a 3' and a 5'. You can abbreviate the strands by just naming the bases that are on them with their letters. So this is A, C, G, T. I don't know if that's what I put here or not. No, it's just a different one. So here, I just put together a sample one of A, T, C, C, T, A, A, G, C, with this being the 3' and this being the 5'. The convention is to list the 5' on the left and lay this out. And then every strand then does have what's called a complementary strand. And what you do is you just simply flip this around, and then you match up the letters differently. So A's and T's always go together, and we're going to see why in a minute. A and T's go together, C and G's go together, so forth and so on. And so the complementary strand of this is this flipped, and then the complementary base being listed here. Okay, so DNA, as we all probably know, DNA has this double helix structure like this. The reason why it has this structure is there is hydrogen bonding between these bases inside this. These are these bases hanging off of that structure that's built together. But you do get some stability from what's called base stacking. So what you have here is, remember, you've got bases in here. So this is our double helix. And here's our bases inside of here, and they stack on top of each other like this in parallel to each other. And therefore that provides some attraction between these and provides additional stability for this double helix structure. Also, these bases are hydrophobic, so they're going to want to stay inside of that helix. And then there are also London dispersion forces that create some attractions in here as well. And I think I have a little thing of this. I don't remember if we talked about London dispersion forces before. This is the weakest attraction force. And this is caused by where two atoms come close together. you're going to have the electrons of one atom are going to be drawn to the protons of another atom, because these are negatively charged and the proton is positively charged. But then the electrons in this atom are going to want to go away from these electrons. And so you get a very brief dipole here. You're going to have a partial positive here, partial negative here, partial positive here, partial negative here. But these are very short and don't last very long, and that's why they're the weakest force. But they do contribute to the overall, the overall structure of the double helix. There are always equal amounts of A and T and C and G. They run anti-parallel. One strand is going to run from 3 to 5, and the other one is going to run from 5 to 3 in order to be a complementary set. There is no hydrogen bonding going on in here, and the reason why is that if you had two purine derivatives in here, they're too large and they would overlap with one another. And if you had two pyrimidine derivatives in here, then they would be too small and therefore too far apart to have any hydrogen bonding between them. And this is why you always are going to get pairs of A and T and C and T in DNA, is that these are, one is purine derivative and the other one is a pyrimidine derivative. And they can do it. You'll never get them between two purines or two pyrimidines. And these are complementary, because if you know one side, you can deduce the other, because you know if this is AAG, this has to be TTC. Okay, DNA replication. What happens when your body wants to replicate DNA is it uses an enzyme called DNA helicase, which unzips the double helix, exposing these active groups here, these base groups. They will then pair with free nucleotides. So you're going to get your G to C and T and A pairings going on. And then another enzyme called DNA polymerase. So polymerase, it makes a polymer. It verifies these base pairings and zips them up. And what you ultimately end up with is two daughter DNA molecules that are like the parent DNA molecule. So here you see the DNA starts to unzip. It starts to create the complementary strand with this. And then that continues as it unzips. And then you end up with two strands that are identical to this original one. Any questions there? Chromosomes. DNA interacts with proteins, which are called histones, to be more stable, and these are called chromosomes. Chromosomes are made up of about 15% DNA and about 85% protein. And they always exist in match pairs. Humans have 46, mosquitoes only have 6, frogs have 26, and dogs have more than humans. They have 78 chromosomes. And one chromosome is always going to come from the father and one from the mother. This is how we get genetic diversity from one generation to another. RNA. So RNA is going to have a ribose sugar, not a deoxyribose sugar. RNA is not in a double helix structure. It's a single strand. It is much smaller than DNA. RNA is usually 75 to maybe a few thousand nucleotides, whereas DNA can have literally hundreds of thousands of nucleotides. But it can double back on itself and be held together with hydrogen bonding. So you do get RNA having a structure like this, where you've got a loop here, and then it comes back. Again, this will become very important in a minute when we talk about protein synthesis, but we have this, what's called a hairpin loop on one end of the RNA strand. There are 5 types of RNA. There's heterogeneous nuclear RNA. This gets abbreviated as HNRNA. This is formed from DNA and then gets converted to messenger RNA, which is the next one on the list. Messenger RNA carries instructions for protein synthesis from the DNA to the synthesis sites where the protein will be made. So we'll see this in a second. Small nuclear RNA gets abbreviated snRNA, and this facilitates the conversion of the heterogeneous nuclear RNA to messenger RNA. Ribosomal RNA combines with proteins to form ribosomes, and ribosomes are the sites where protein synthesis occurs. And then finally, we have transfer RNA, abbreviated tRNA, which transfers amino acids to these protein synthesis sites. So what happens is we have an RNA polymerase, which unwinds a portion of the DNA, and the free nucleotides then come in and bond. But because it's RNA, you get UA linkages and CG linkages. And then the RNA polymerase encounters a stop sequence, which we'll see in a moment, and stops doing the combinations. And then the RNA polymerase releases the RNA, and then the DNA rewinds itself back up. So we have a DNA here, it gets partially unwound, then it comes in and transcribes, makes a copy of just this one section here, and then the DNA winds back up and we're left with a RNA, and this is the heterogeneous nuclear RNA here. A good way to remember this is nuclear is like nucleus, which is where the DNA is at. So here, if your DNA template is ATGCCA, then your RNA molecule that comes from this is going to be the flip of that, and then the complementary bases. And so you're going to get AU, CG, 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 TAAU. So this is the complementary RNA molecule that will produce from that. Then what happens is the formation of the messenger RNA. Not all segments of a DNA carry genetic information. They actually don't know why this is, but my guess is it has to do with protecting from mutations and that kind of a thing, but I'm no biologist, I don't know. Exons are sections of DNA that have genetic material, and introns do not. And so what happens is that the HNRNA gets the introns are removed and the messenger RNA is just the exons that are left over. So if you have this hnRNA strand that came from your DNA, if these two were introns, they get pulled out, and then these three just get merged together, and now you only have genetic instructions in that messenger RNA. Codons are 3 nucleotides that provide information for the building of amino acids. Codons are degenerative. This means that multiple codons will specify to build the same amino acid. And these are called synonyms because they're building the same one. So you can see here we've got a codon of these 3 nucleotides, A, U, and G. We got another one here of G, G, and U. We've got another one here of G, C, and U. This yellow here is that ribosome, and this is where it's going to start building the protein based on these instructions from these codons. And so the way to read this chart is this tells you, I'm not going to ask you to memorize how, this chart or anything. I'll give it to you on the test, but then ask you to utilize it. But the way to read this is that you look at the first, second, and third position of the codon. So if our codon is ACG, then you look at the first position A, the second position is C in this column, but then you look at the third position is G in this row with the A, and draw over, and you see that that ACG is an instruction for building threonine here. See how that works? You guys familiar with this? You've seen this before?
Yeah.
Okay. If you had GUG, then you go to your G is your first one, then you use this column for the U, and then this row that the G is in for your G, and this tells you that GUG is a instruction to build valine. There is one in codon which tells it to start building. This is the AUG so it'd be AUG is this methionine. It also means to start. And then there are 3 codons that indicate an int to stop building. So it will run until it hits these codons, one of these codons, and then it will stop building. And then here's an example of some synonyms. All 6 of these codons are an instruction for building arginine. So if you look at C, G, C, you get to arginine. And if you look at C, G, U, you also get to arginine. So all 6 of these are synonyms. They build the same thing. So this is where that hairpin becomes important, is transfer RNA has what's called an anticodon in that hairpin. And on the opposite end, you will get attached there the opposite of this CUU. In this example, would be GAA. And GAA is an instruction to build glutamic acid. So glutamic acid will attach here on this end, and this gets used as the mechanism for building a sequence of proteins. So what we see here is, here's this yellow is this ribosome site. There's two sites within a ribosome. There's the P site and the A site. So we start on the P site, and you get this AUG to say start building. And so it comes in here, and it's going to have the anticodon for this is going to be UAC. So that's going to be drawn in and connected here. And this is going to have methionine on that end. So that gets attached. And let's see, there's that. And then you're going to have the next one come in. So this one moves. I don't think this shows us quite right, but this one would start here and then move to the P site. And then now you would have this one come in. In this example, we've got GGU. The anticodon is CCA. And this is an instruction for glycine. So this is attached to this end of it. So these are both now in the ribosome. And then what happens is that the methionine connects to the glycine with a peptide bond. And then this transfer RNA is going to leave the ribosome. This will then shift over to the P site, and your next one will come in. And so here we have GCU. The anticodon is CGA. This is for alanine. So this comes in, and then it's going to connect to this as well. And this will keep running until it hits a codon that says stop. So here we have the alanine now combining. So we've got this sequence of these three together. And then the next one is going to come in for phenylalanine. And then it's going to go until it hits either UAA, UAG, or UGA. And this process is called elongation, where you're building a longer protein by combining these amino acids together. Most proteins do not have a methionine as their first amino acid. So this is, it gets used as a start codon. It usually gets cleaved off. So this very first one in post-processing gets removed because it was this just there to start it all off. And sometimes in post-processing, you'll get disulfide bonds forming between any cysteine units that are in the chain. The transcription phase is where the heterogeneous nuclear RNA gets formed. The introns that do not have genetic information gets removed and makes the mRNA. This is called the transcription phase for this. And then the mRNA leaves the nucleus and goes out into the cytoplasm of the cell, where it can be used in the ribosome. The translation phase then is the activation of the transfer RNA. You get the amino acid bonding to the 3' end of that transfer RNA. The messenger RNA attaches to the ribosome so that the first codon is on the P site. Another one attaches to the A site. The amino acids bond together, and then the process continues until you hit a stop codon, and then the new protein is cleaved off and you get the post-translation occurring. So here it is all together with a hypothetical one. If we had a DNA strand here with these nucleotides, And the exons are A, C, and E. So those are the ones that have meaningful genetic information. You're going to get your hnRNA is going to be the complementary strand to this. So you're going to get it looking like this here, where you've got GGC to match up with the CCG, then ACU to match up with the TGA, so forth and so on. But then your messenger RNA is where these introns here get removed, and you just have the meaningful information left over. And then this is going to build a protein which is made up of these amino acids. Any questions there at all? Again, probably mostly review for you guys, right? Mutations can occur in this process. Mutagens are substances or agents that cause damage to the gene structure. This can happen with ultraviolet radiation, X-rays, radioactivity. Also, the presence of nitrous acid can cause these. I got an example here where nitrous acid will actually change cytosine to uracil. So you've now got a codon that's changed, and this is a mutation. So instead of having CGG, you now have UGG. Sorry, I'm kind of losing my voice. The sad news is that the food industry uses preservatives to try to make their food last longer on the shelf. And a byproduct of that is the presence of nitrous acid. And so you can get mutagens formed or mutations formed from those food preservatives. There are repair enzymes which will attempt to repair the affected bases, but if they're unable to do it, then the mutation will persist. Viruses have DNA or RNA that cannot reproduce without a host cell. So they have to be in a host cell in order to reproduce. Therefore, this means they're not really alive. Viruses are not alive because they can't reproduce on their own. They have to have this host cell to do it. Viruses not only affect humans, but also animals and even plants can contract viruses. Some common examples are the cold, mumps, measles, smallpox, rabies, the flu, hepatitis, and AIDS. These are all viruses. Probably most of you guys are young enough, you never had mumps or measles. Anybody have mumps or measles when they were young? I had both when I was a kid. But there's vaccines, so they're not very common these days anymore. If the virus has DNA, then it hijacks the replication process that we just looked at in order to make its own proteins. And if the virus has RNA, this is called a retrovirus, and it makes viral DNA, which then makes proteins. AIDS is an example of a retrovirus. Vaccines have inactive or weakened forms of the virus, and this allows the body to build up antibodies to fight them off. Oops, going the wrong way. Any questions there at all? Okay, have you guys studied recombinant DNA before? I found this very fascinating when I first learned about this. So, what scientists figured out is that they can reproduce a strand of DNA that they're interested in studying. What they do is they introduce two restriction enzymes that will take off a piece of this DNA, this section that they're interested in, and then they insert it into a plasmid. Plasmid is a circular strand of DNA. So they'll take this piece off of here and put it into a plasmid using a DNA ligase. Remember, ligase creates bonds. So this DNA ligase will graft this in here. They will also then add to the plasmid a antibiotic resistance chemical. And then what they do is they culture these in E. coli cells are commonly used, or sometimes yeast. And then what they do with that culture is they treat it with a antibiotic, and it will kill off the ones that don't have this antibiotic resistant strand in them. And so you're left with just the ones that have the DNA that they're interested in. So this kind of acts like a host cell, if you will. We just kill off the ones we're not really interested in. And then now you can bring out of there the replicated genes that are going to be identical to this section of the DNA. Any questions there? I found that pretty cool that they figured out how to do this. And this is the two strands of DNA. They would be wound together. I just drew them separately to illustrate here. A polymerase chain reaction, abbreviated PCR, is a way for scientists to take a few molecules of a DNA and multiply it out. And this quite often gets used like in for diagnoses, because you multiply it so it's easier to measure. It gets used in detecting AIDS. It also gets used in forensics. So if you just have one strand of hair or something, or one little piece of a bone, they might be able to get some DNA out of there, but it's not enough to measure. So they'll run it through this PCR reaction in order to multiply this DNA out into a large amount so that they can then measure it more easily. And how they do this is they take the DNA strand, they heat it up to about 96 degrees Celsius, which is pretty hot, and this will break the strand apart. They will then cool it down to about 55 degrees Celsius, and they will add in nucleotides that are called primers, and they will attach to these strands. And then and this is a process called annealing, and then they will heat it up again a little bit more, not all the way to 96, but to 72 degrees Celsius. And then what happens is they have a polymerase in there. This is called a Taq polymerase, which will operate at these high temperatures, and it will create the complementary strand, and so you've now multiplied this. So you've gone from one strand of DNA to two, and then they repeat this process over and over again. So in solution, you have the DNA you're interested in, this Taq polymerase, You're going to have free nucleotides so that they're available to bond in here and form these strands. And then you'll have water and some nutrients as well. And they'll run this like perhaps 35 different cycles. And so when you do that, you will get 2 to the 35th copies of this original DNA strand. And these days, this only takes about 2 to 3 hours to do. And so you can see 2 to the 35th power is a huge number. So they've really blown out and multiplied this. And you now have a lot more DNA that's easier to read than just a few molecules of it. The interesting thing is they needed a polymerase that could survive at these hotter temperatures. And where they found it was at Yellowstone. This TAC actually stands for Thermus aquaticus, which is the name of this microbe that lived in the hot water in the hot springs at Yellowstone. And they found this enzyme in that. And those microbes were able to survive in that great heat. And part of the reason why is that they had an enzyme that could survive in that great heat. And so they used this in this process because they needed to heat up in order to make the process happen. Pretty smart, huh? Just pretty crazy they figure this stuff out. DNA sequencing uses PCR. So if you're wanting to know what the sequences are in this DNA strand, then the first thing you do is you multiply it out to make sure you have enough to do. But what you do is you add nucleotides that have a fluorescent chemical attached to them. And then what happens is you heat and cool this repeatedly, and you will get strands breaking up as a part of this heating and cooling process. And you'll get some that only have 2 nucleotides, some that only have 3, some that only have 4, so forth and so on. And only one of these will be fluorescent. So only one of the nucleotides will be fluorescent. And then what they do is they pass this through a gel with an electric current. And what this does is it causes the strands to separate out. And you will get the shorter strands first, and then you get the longer strands, second, third, fourth, however many. And now as these leave, as they're coming through this gel, they have a laser here that will read. It will cause these strands to fluoresce, and then it will tell you, I've got the first one is an A, the second one is a C, and the third one is a T, because it's reading these one group at a time as they're coming through. Does that make sense? Do I have a picture of one? No, I guess not. And so this is a way that they can get the sequence of the DNA is through this process of DNA sequencing using fluorescent markers. Any questions there at all? And like I said, it was a shorter chapter, so we are at the end. I put some problems in here, depending on 4th or 7th edition, that you can look at. Next week, we will do a review for exam 5. Exam 5 will cover chapters 9, 10, and 11. And that exam is not this weekend, it'll be the following weekend. So we'll have a time to do a review next week. And then we will also cover chapter 12 next week. So, any questions, anything at all before we wrap up? If you want to stay and